plant health and agricultural productivity. Elements such as cadmium, lead, mercury, arsenic, chromium, and nickel accumulate in soil due to industrial activities, mining operations, and excessive use of chemical fertilizers. Heavy metal stress disturb multiple cellular processes leading to physiological and biochemical damage. One of the most critical consequences of heavy metal exposure is, of course, oxidative stress. Heavy metal interfere with electron transport in mitochondria and the chloroplast, causing excessive production of reactive oxygen species. These highly reactive molecules damage cellular membranes, proteins, DNA, leading to cellular dysfunction and ultimately cell death. Another significant impact is photosynthetic inhibition. Heavy metals damage photosystem II, which plays a vital role in light-dependent reaction on photosynthesis. On the previous lecture, Professor Shamsul discussed about the role of phytohormones, the role of brasinosteroid on photosynthesis, and told about the fundamental role of photosynthesis in plants. However, in the results when the plant are treated with heavy metals is a decline in chlorophyll contents and reduced energy production, severely affected plant growth and development. Moreover, heavy metals induce a nutrient imbalance by interfering with the uptake of essential elements such as calcium, magnesium, zinc, or iron. These nutrients, of course, are very important elements for enzymatic reactions, signal transduction, and overall plant metabolism. When they absorb in hindered key physiological processes, including protein photosynthesis or enzyme function, became compromised. Additionally, heavy metals can directly interact with DNA and proteins, binding to sulfhydric thiols groups of enzymes and structural proteins, and this disturb gene expression, enzyme activities, and metabolic pathways, further intensifying the stress response in plants. As a result of this disturbance, plants struggle to maintain normal growth and survival in contaminated environments. To better understand this effect, we can employ various biochemical spectroscopic and molecular techniques to analyze stress markers and evaluate plant responses to heavy metals. In addition to the oxidative stress and the nutrients imbalance, heavy metals toxicity significantly affects plant hormones, of course, which are critical regulators of growth, development, and stress adaptation. We know everything about action of phytohormones and classes and signal transductions and biochemical action in plants. However, for example, auxin is often reduced under heavy metal stress, since auxin regulate root and shoot development. Their decline leads to inhibited root elongation, stuntman growth, and impaired cell division. Consequently, abscisic acid, as we know as a stress response hormone, is typically upregulated in response to metal exposure. Abscisic acid helps plant cope with stress to closing stomata to reduce water loss and triggering defense mechanism. However, excessive ABA accumulation can lead to growth retardation and reduce photosynthesis. Similarly, gibberellins, which promote stem elongation and seed germination, are often down-regulated under stress, leading to reduced growth and developmental delay. On the other hand, jasmonic acid and salicylic acid, which are stress-related hormone, increase under metal toxicity. This hormone activate plant defense responses, including antioxidant production and metal detoxification mechanism. Understanding how heavy metals influence these hormonal pathways is essential for assessing plant stress responses and adaptation strategies. So one of the primary indicators of oxidative stress is the accumulation of reactive oxygen species, such as superoxide radicals and hydrogen peroxide. These molecules damage lipid, proteins, and DNA, leading to impaired cellular function. To counteract oxidative stress, a plant activates the antioxidant defense system, which include both enzymatic and non-enzymatic antioxidant. Enzymes such as superoxide dismutase, catalase, and peroxidase play a critical role in neutralizing reactive oxygen species. Meanwhile, non-enzymatic antioxidants like glutathione, ascorbic acid, and phytogelatin help detoxify heavy metals and maintain cellular redox balance. One of the most significant biochemical markers of oxidative stress is lipid peroxidation. This process occurs when reactive oxygen species attack polyunsaturated fatty acids in cell membranes, leading to structural damage and loss of membrane integrity. The primary byproduct of lipid peroxidation is malondialdehyde, MDA. It serves as a reliable indicator of oxidative stress levels in plants exposed to heavy metals. So, how does this process occur? Under metal stress, reactive oxygen species such as superoxide, hydrogen peroxide, and hydroxyl radicals accumulate in plant cell. These reactive oxygen species react with membrane lipids, initiating a chain reaction that leads to the breakdown of lipid molecules and the formation of malondialdehyde. The significance of MDA accumulation lies in its direct correlation with membrane damage. Higher MDA levels indicate greater oxidative damage, resulting in disturbed cellular function, impaired water and nutrient transport, and ultimately reduce plant growth. To quantify MDA level, we can use the thiobarbituric acid test. In this assay, MDA react with thiobarbituric acid to form a pink color complex, which can measure using spectrophotometers at 500 and 32 nanometers. This method provides a precise and reliable measure of lipid peroxidation in stressed plants. This is the first method. Spectrophotometrically we can measure stress markers. The first stress markers is MDA, MDA malonyl dialdehyde. In addition to lipid peroxidation, another important indicator of cellular damage under heavy metal stress is electrolyte leakage. This test assays the integrity of plant cells membrane by measuring the count of ions such as potassium, calcium, and chloride that leak out of the damaged cell. Under normal conditions, plant cell membrane as a selective barriers regulated ion transport and maintaining cellular homeostasis. However, when plants are exposed to heavy metals, this toxic ions disturb membrane stability, causing structural damage and increased permeability. As a result, essential ions leak out of the cell, indicating loss of the membrane function. So, how do we measure electrolyte leakage? The electrolytic conductivity method is commonly, of course, used. In this assay, no. Where's in this assay, leaf discs of root segmentation are collected from the plant. The sample are incubated in distilled water, allowing the ions to diffuse out. The electrical conductivity of the solution is measured before and after autoclaving. Autoclaving completely disturbs the cell membrane, releasing all intracellular ions. The percentage of electrolytic leakage is then calculated as an indicator of membrane damage. Higher electrolyte leakage values correspond to the greater cell membrane injury, reflecting severe heavy metals toxicity. This test provides a rapid and effective way to access early sign of stress, helping us to evaluate plant tolerance and tendency to metal contamination. By combining electrolyte leakage measurements with lipid peroxidation assay, we can gain a comprehensive understanding of how heavy metals impact plant health at the cellular level. Reactive oxygen species, one of the most, maybe most significant produced in plants, superoxide. This highly reactive molecules is primarily formed in the mitochondria and the chloroplast when heavy metals disturb electron transport processes. How is superoxide generated? It occurs when molecular oxygen gains an extra electron, often due to the interference of metals with electron transport chain. The reaction is follows. Another major source of superoxide is NADPH oxidase, an enzyme that generates superoxide as a part of the plant stress response. While lower levels of superoxide are essential for signaling and defense, excessive accumulation leads to oxidative stress. To quantify superoxide production, we commonly use NBT staining, that's mean nitro blue tetrazolium assay. In this method, plant tissue are incubated with NBT, a dye that reaction with superoxide. Superoxide reduce NBT to form blue formazan precipitate, which are then visualized and quantified. The intensity of the blue coloration correlates with superoxide levels, allowing researchers to access oxidative stress severity. By monitoring superoxide level, we can evaluate the extent of reactive oxygen species-mediated damage and determine how efficiently plants activate the antioxidant defense mechanism. The hydrogen peroxide is a here reactive oxygen species involved in heavy metal stress. Unlike superoxide, hydrogen peroxide is relatively stable and can diffuse across membrane, making it a crucial signaling in plant stress responses. Hydrogen peroxide is primarily formed through the enzymatic conversion, superoxide by superoxide dismutase, SOD. The reaction follows this, in this pathway. While H2O2 plays a beneficial role in activating stress response genes, excessive accumulation leads to oxidative damage, lipid peroxidation, and protein denaturation. It makes it is a critical biomarkers for evaluating oxidative stress levels in plant exposed, of course, to heavy metals. To quantify hydrogen peroxide in plant tissues, we use colorimetric and histochemical assays such as potassium iodide, starch colorimetric reaction. It means that hydrogen peroxide react with potassium iodide, producing a yellowish brown color that can be measured spectrophotometrically. In my lab, of course, this test and the method is commonly used. By measuring hydrogen peroxide level, we can assess the balance between oxidative stress and antioxidant defense mechanism in plants. Understanding this balance is essential for identifying stress-resistant plant species and improving, for example, growth or plant tolerance to heavy metal contamination. To maintain cellular homeostasis, plants rely on antioxidant defense systems, which include enzymatic antioxidant. I said about superoxide dismutase as SOD, catalase CAT, and peroxidase POD, which neutralize reactive oxygen species. The next are non-enzymatic antioxidants such as glutathione, ascorbic acid, and phytogelatins, which of course help detoxify reactive oxygen species and heavy metals, especially phytogelatins. By quantifying ROS levels and analyzing antioxidant activity, we can better understand how plants respond to heavy metal stress and develop strategies to enhance their resilience and, of course, detoxification by using, by production, by synthesis, phytogelatins. As we have seen, plant response to heavy metal stress production large amounts of reactive oxygen species, which can cause several oxidative damage. Three majors, of course, I said about S of D, superoxide dismutase, which converts superoxide into hydrogen peroxide and oxygen, preventing excessive ROS accumulation. Catalyze break down hydrogen peroxide into water and oxygen protection cell from oxidative stress. Peroxidases catalyze the reduction of hydrogen peroxide by oxidizing various substances, helping detoxify reactive oxygen species. It's, of course, very, very easy. By measuring the activity of this enzyme, we can determine how efficiently plants are responsive to metal-induced stress. Increases enzyme activity indicate that the plant is activity is scavenging reactive oxygen species, while reduced activity, it suggests, of course, that oxidative stress may be overwhelming the plant's defense system. To quantify antioxidant enzyme activity, we use spectrophotometric assay based on specific reactions of this enzyme with the substrate. The enzyme activity is typically expressed as enzyme unit per milligram of protein, providing the measure of antioxidant capacity. Of course, we must measure the total level of protein. Antioxidant procedure is very, very easy. In many publications, there are in detail present. In addition to enzymatic and non-enzymatic antioxidant, plants have a special mechanism for detoxifying heavy metals. It is phytohalatins. What are phytohalatins? They are small cysteine-rich peptides derived from glutathione. They bind to heavy metals, such as, of course, cadmium, arsenic or lead, to form a PC, heavy metal, phytohalatin metal complexes, which are then substrated into vacuoles, reducing metal toxicity in the cytoplasm. General reaction of phytohalatin metal complex formation is presented. Why are phytohalatins important? Because they prevent heavy metals ion from interaction with essential cellular components. They enhance metal sequestration into vacuoles, reducing cytoplasmic toxicity. And their production is regulated by enzyme phytohalatin synthase, is important to metal exposure. How are phytohalatins quantified? The measurements is analyzed by HPLC with fluorescence detection. I don't know about other methods. We use in my lab only HPLC with FLD detectors. The steps include extraction of phytohalatins from plant tissue and, of course, derivatization with mono-bromo-biman which enchants fluorescence. It is the only one derivatizator commonly used and commonly applied. Separation and then separation is very easy separation, of course, similar like to phytohormones, purification and then detection using only HPLC systems. The long-term experiments show that, for example, phytohalogens PC2 is the mean dimer, cis and glue is than 3 times, 4 times, maybe 6 times. Theoretically, the dimer is from 2 to 10, but usually, maximally, we measure only phytohalogens 5 in the long-term experiments. By quantifying phytohalogens levels, we can assess a plant's ability to detoxifying heavy metals and identify species with strong metal tolerance. This information is valuable for developing crops that can thrive in contaminated environmental or be used for phytoremediation. In summary, biochemical assays provide quantitative insight into plant stress responses, particularly under heavy metal exposure. By analyzing oxidative stress markers and antioxidant defense system, by, of course, non-enzymatic and enzymatic antioxidants, we can better understand how plants cope with heavy metal toxicity. Key takeaways may include oxidative stress and reactive oxygen species generation. That means heavy metals induce excessive reactive oxygen species such as superoxide and hydrogen peroxide. And reactive oxygen species could cause lipid peroxidation. Is measured, of course, by MDA malondialdehyde, leading to the membrane damage and increased MDA levels. Electrolyte leakage is a key physiological indicator of membrane integrity loss. And then, as of course I said about antioxidant defense mechanism is by enzymatic antioxidant, which play a crucial role in reactive oxygen detoxification, and non-enzymatic antioxidant glutathione and ascorbic acid, which of course that we can measure by HPLC. They maintain redox balance and support enzymatic activity. And the next heavy metal detoxification by phytohalogens, which bind heavy metal and facilitate the sequestration into vacuoles and reducing toxicity of heavy metals. This detoxification mechanism determines plant tolerance levels and survival under heavy metal stress. Why are these assays important? The first, of course, they help identify stress tolerance plant species. They support breeding programs for metal resistant crop. They contribute to phytoremediation strategies using plants to clean contaminated soils. Of course, not soils, maybe of water. In addition to biochemical assays, advanced spectroscopic and chromatographic techniques play a crucial role in analyzing plant stress markers under heavy metal exposure. This method provides high precision data on metal accumulation in plant tissue, oxidative stress markers, and phytohormonal balances, which regulates plant growth and stress responses. The key techniques used in the field is, of course, the HPLC used to quantify antioxidant and other secondary metabolites. GCMS identifies volatile organic compounds and lipid peroxidation byproducts. And ICP-MS, ICP-MS, which measure heavy metal concentration with high sensitivity, is a better instrument than AS, and the Fourier transform infrared spectroscopy, which detects biochemical changes in protein, lipids, and carbohydrates. One of the most used techniques for plant stress analysis is HPLC. Why HPLC is important? It's essential for studying heavy metal response because it helps measure antioxidant compounds, including glutathione. I said about it, and phytohalatins, which help detoxify metals. How does HPLC work in these measurements? Sample extraction, plants extract containing target compounds are prepared, separation on HPLC column, typical C18. The sample passes through a column filled with stationary phase, mobile phase in methanol. Different molecules are separated based on the chemical properties and the detection and quantification. Of course, we must have fluorescent detectors, which is used for glutathione and phytohalatin analysis. When you use UV and mass spectrometry, we can detect, of course, plant hormones and oxidative stress markers. About detection of plant hormones, I said in the previous lecture, I don't remember when. Another powerful tool for studying plant responses to heavy metal stress is GSMS. This technique is particularly useful for identifying and quantifying volatile organic compounds. In the part, of course, phytohormones and lipid peroxidation byproducts. GCMS play an important role in plant stress research by detecting volatile phytohormones such as ethylene, methyl yasmonates, and abscisic acid, which regulates stress response. Analyze lipid peroxidation byproducts such as melatonin dealdehyde and other, which indicate oxidative damage. Identify secondary metabolites such as terpenoid or alkaloid that contribute to plant defense mechanism. So the application of GCMS in heavy metal stress research can evaluate oxidative stress markers by detecting lipid peroxidation byproducts, and studying hormonal regulation by measuring stress-related volatiles, assessing plant metabolism, and identifying key biochemical changes under metal toxicity. I can use this metal molecular methods in my lab, so I would like to omit. Imaging and microscopy techniques provide visual insight into how heavy metal stress affect plant cell. These methods allow researchers to observe structural damage, metal localization, and oxidative stress responses at cellular and subcellular levels. The key microscopy techniques used in heavy metal stress as a biomarkers research include fluorescence microscopy, confocal laser scanning microscopy, scanning electron microscopy, transmission electron microscopy, and X-ray fluorescence microscopy. The first detect reactive oxygen species accumulation and stress responsive using specific fluorescence dyes. The next confocal laser scanning microscopy provides a high resolution image of heavy metals distribution and antioxidative stress markers. On the other hand, scanning electron microscopy examines surface damage in levels root and stems. Transmission electron microscopy (TEMM) reveals ultrastructural changes such as chloroplast degradation and vascular sequestration of metals. And the last, X-ray fluorescence microscopy, maps the localization of heavy metals within plant tissue. Let's go to conclusion. Let's reflect on the biggest picture. How do plants survive in metal contaminated environments, and how we can learn from them. Heavy metal stress is a silent threat to agriculture and ecosystem. Heavy metals such as cadmium, lead, mercury, arsenic don't just accumulate in soil. They invade the plant cells, disturb metabolism, and threaten food security. Yet plants have enveloped an arsenal of defense mechanisms to fight back. What have we discovered? The first, of course, the heavy metal trigger a storm of reactive oxygen species, damaging protein, DNA, and membranes. They hijack nutrients transport system, leading to defenses in calcium, zinc, and iron. They disturb, of course, heavy metals, photosynthesis, hormone balances, stunting growth, and lowering yields. Antioxidant various, for example, SOD, CAT, and POD, neutralize reactive oxygen species before they cause irreversible damage. Molecular mechanism system such as glutathione, ascorbic acid, phytogelatins trap and detoxify heavy metals. Gene activation plants fine-tune their stress response in real time. And the next, of course, HPLC, GCMS methods, techniques reveal the hidden changes in stress metabolism. ICP-MS, for example, map heavy metal accumulation at the atomic level. Fluorescence and electron microscopy give us a close-up view of plant cells fighting for survival. Questions. Can we engineer crop to be more resistant to plant metals? The answer is yes. How can we use plant to clean the contaminated soils through phytoremediation? Please answer yourself. Plants have been adapting to stress for a million years now. It's our turn to learn from them. By integrating biochemical, molecular, and imaging techniques, we can develop sustainable strategies for protein crops, restoring ecosystem, and ensuring global food security. Thank you very much.
Thank you, Professor, for your nice lecture. The next lecture